Well, hello. This is the first team-based learning section that you will be responsible for on exam 1. So make sure you all um, go through these slides, go through this YouTube video, and take a close look, and make sure you got all the information that I give you on these slides. And this is what we're going to be covering in class on Monday. We'll go ahead and start. Okay, so the body planes, you guys have already studied this in lab, and you've studied it in the other handout in the YouTube video that I posted. But we'll just quickly show one thing I want you to focus on is knowing what a movement in a plane actually means. Okay, so let's look at the sagittal plane for that. Okay, so we're over here looking at the sagittal plane. So sagittal plane movement would be like flexion and extension of the elbow. Okay, so just start in an anatomical position, flexing the elbow, coming straight up this way. So movement within that plane. Another movement would be nodding your head, as in you're saying yes. That's another sagittal plane movement. Okay, it's important to know the difference between mid-sagittal plane and parasagittal plane. The mid-sagittal plane is the one directly in the center of the body. But you can also have other sagittal planes which are not in the center, but they are in that same plane. Okay, next is the frontal plane, also known as the coronal plane, and so it's important to know that some movements that happen in this plane. So if you were to take your head and take your ear and move it all the way to your shoulder. So your left ear, go down and touch your left shoulder, and then go back right ear to the right shoulder. That's a movement in the sagittal plane. Also shoulder abduction, which would be moving the shoulder up this way. Hip abduction and adduction, and those are all movements in the in the frontal plane. Okay, and then finally is the transverse plane, and this is kind of known as a rotational plane. So movements in the transverse plane include rotation of your neck. So think of in this case saying no. So nodding your head in the fashion to say no would be a transverse plane movement. Okay, as well as rotating your hips. So put your hands on, just like that stretch you do, put your hands on your hips and rotate your body one direction. That's a transverse plane movement. Hip rotation, shoulder rotation, wrist pronation and supination, those are all transverse plane movements. Okay, I'm not going to talk about these, but these are all terms that you need to know. I think you already know them already from lab, but make sure you know these for the exam. All right, okay. One thing that we didn't cover in class is looking into a little bit of detail on all the different organelles inside of a cell. Okay, so organelles, the definition of an organelle is a small component that's found within a cell, and they all have different functions. So first one is the nucleus. It's the center of the cell. It's the site of all hereditary material, and that's the DNA. Okay, and this DNA is what encodes for all proteins that make up most of the body. Okay. And so that's all found in the nucleus. This is the largest organelle in the cell. Okay, another very important organelle in the cell is called the mitochondria. Okay, and the mitochondria's main function is to produce ATP. ATP is adenosine triphosphate, and this is the source of energy that all cells use to do all cellular functions. Okay, so within the mitochondria, there's the inner membrane, which is this, and there's the outer membrane. Okay. You see inside the mitochondria there's lots and lots of folds. Okay, this inner membrane is just folded like crazy, and those folds are called cristae. Okay, the reason for this folding that you see here is to increase surface area. Okay, the more folds we have, then the more surface area we have, and the reason we want surface area inside the mitochondria is because the electron transport chain, which we'll talk about, happens by ions and different molecules passing back and forth across this membrane. So the more membrane surface area we have, the more ability we have to do the electron transport chain, and that's how we make ATP for the most part. Moving on. Other important organelles are called ribosomes. Ribosomes are made up of protein and RNA. Okay, these are known as protein factories. So these are the organelles that make proteins. Okay, so they take messenger RNA that started in the nucleus and translate that into amino acids, which and then they help to put those amino acids together and form proteins. Okay, so some of these ribosomes are bound to the rough endoplasmic reticulum, which is shown right here, and that's actually what makes it rough, is the ribosome sticking out. And then some are just floating freely, and both of them are there to help us make proteins. Okay, next, this is kind of not necessarily a single organelle, but this is what's called the endomembrane, which is a system of linked organelles. Okay, the main function of this endomembrane is to produce, store, and export molecules. Okay, so for example, we start with creating a protein inside the nucleus. Okay, so let's say we're inside of a pancreatic beta cell. Pancreatic beta cells are the cells that make insulin. Okay, which is an important hormone for controlling blood sugar. Okay, so the insulin's a protein, and we start by decoding the DNA to get the right amino acids. Okay, so the first part of the endomembrane is in the nucleus. Okay, and then the next part is the rough endoplasmic reticulum, where that protein is formed even more, and then from there we go into the Golgi apparatus, which is shown here in green, and that's where we process the proteins, and then the Golgi apparatus splits off into a little circle here, which is shown here, which is a transport vesicle, and then that insulin that was just created can be stored there, and then when it's called to be released, that vesicle can go to the surface of the cell and release the insulin into the cell, or outside of the cell, I'm sorry. Okay, and that's the main idea there with the endomembrane. Okay, next important organelle is called the rough endoplasmic reticulum. Okay, like I said, it's rough because it's covered in ribosomes. Okay, this is the closest organelle to the nucleus. You can see here, this is the nucleus. There's these nuclear pores which connect right to the rough endoplasmic reticulum. Okay, functions are creating proteins. So the ribosomes produce proteins. These proteins are packaged into vesicles, and then these vesicles are formed by endocytosis from the rough endoplasmic reticulum. Part of it just comes off and forms a vesicle. Okay, this vesicle carries proteins to the Golgi apparatus, and then the vesicle empties its contents into the Golgi apparatus by exocytosis. Okay, so the main key here is the function, and that's the ribosomes are there to produce proteins. Okay, next is the smooth endoplasmic reticulum. This has nothing to do with making proteins. This has everything to do with making other things, and some of those things are lipids. Okay, so there's a series of tubes filled with different types of enzymes. Okay, so the main function here is to manufacture lipids, including cholesterol. Okay, so these help us produce steroid hormones. Steroid hormones are hormones that have a cholesterol base, cholesterol backbone, we should say. Inside the intestinal cells, we have a specialized smooth endoplasmic reticulum that's there to absorb, synthesize, and transport fats. Okay, these are also very helpful. The smooth endoplasmic reticulum is also very helpful in drug detoxification. And then in the liver cells, this is where glycogen is broken down into glucose, so we can use it for energy. In the muscle cells, we have a special form of the smooth endoplasmic reticulum called the sarcoplasmic reticulum. Sarco just means muscle. And that is, we'll talk more about the function of that, but it helps us with muscle contraction. Okay, now we have the Golgi apparatus. It's kind of looks like this here. And the main function of the Golgi apparatus is to modify proteins, kind of putting the finishing touches on the proteins after they go through the rough endoplasmic reticulum. So some examples of this could be add or trim sugars off of the proteins, add a phosphate group, take some amino acids off that aren't actually needed, and some things like that. So. And then once they're done processing, they tag them for 3 different routes. One could be exocytosis. So this, once they're done processing, they go into these vesicles, and that vesicle can be sent immediately to the cell membrane and sent out for exocytosis. Okay. Another option is the vesicle membrane the Golgi apparatus can actually just be sent out to the cell membrane, just to add more cell membrane to the cell. If the cell's growing or if part of the membrane was damaged, the Golgi apparatus could send it out that way. Okay, and sometimes there's proteins inside of those that need to be incorporated into the cell membrane, like those transport proteins we've talked about, or like a certain receptor or something like that. And then last option is this is where lysosomes are made, which we'll talk in a second about what lysosomes are. Okay. So lysosomes and peroxisomes look very similar. They both, the lysosomes bud off of the Golgi apparatus, okay, and they look like these little vesicles, just like they both look similar, just like little vesicles. And these are full of what we call degrading enzymes, so things like hydrogen peroxide, so lots of free radicals, pretty much. Hydrogen peroxide contains lots of free radicals. Okay, and if cells come into contact with these, it destroys that portion of the cell. Okay, and so these are really the main function of these is it's kind of the cell demolition crew. So they can degrade harmful substances. They can degrade the whole cell if needed. If it's time for that cell to die, the lysosomes can just dump their contents out into the center of the cell, and that will destroy the cell. But it can also degrade just portions of the cell. Maybe there's a portion of the Golgi apparatus or a certain protein within the cell that's not functioning anymore. The lysosome will phagocytose or envelop that portion of the cell and destroy it, so it can be replaced later. Peroxisomes are similar to this, but they come off of the endoplasmic reticulum instead of the Golgi apparatus, and these are full of powerful chemicals as well. And these actually are there to quench free radicals. So if we get a lot of free radical release from the mitochondria when we create ATP, and some of those free radicals can be quenched by these peroxisomes kind of enveloping those free radicals. These are very prominent in kidney and liver cells where we have to do lots of drug detoxification. Found in plant cell walls, instead of having a bilipid membrane like we do in our cells, plant cells have cellulose, which is a substance that we can't break down, also known as fiber. Okay, sorry, you already saw that. So glucose is found in the blood. This is an example of what glucose looks like. These are all monosaccharides, okay? You don't need to know the difference between the different monosaccharides, but you do need to be able to recognize a monosaccharide, okay? So it has one carbon ring. So they must all be broken down to monosaccharides for metabolism. So disaccharides and polysaccharides must be broken down into these monosaccharides first before they can be used for energy in the body. Okay, moving on to disaccharides. So these are the examples of disaccharides like I just mentioned. So you can see there's 2 carbon rings in each of these. Okay, so these are your very typical sugars. Okay, so sucrose and fructose are very, very typical sugars. So the early soft drinks were mostly sucrose, which is really just table sugar. Okay, and what sucrose is 40% fructose and 60% glucose. Okay, so that's sucrose right here. Okay, but like this typical sugar was that. But now what we use mostly is high fructose corn syrup, which I'm sure you've heard a lot of hype about. It's really not all that much different from regular old sucrose. It is 45%, either 45% fructose or 50% fructose. So it only shifts by 5 to 10% away from what we knew of as table sugar. And there might be a little bit of extra hype about that high fructose corn syrup that's not really that important. Okay, and then finally the polysaccharides. So this, you need to be able to recognize polysaccharides, and so you can see lots of carbon rings together. Okay, one of the most important ones in the body is glycogen. Okay, and glycogen is important in the body because it's how we store glucose. We can't just store glucose. It's either in the blood or it's being used. And so in order to store it, we have to turn it into glycogen, which is putting a bunch of glucoses together like this. Okay, and it's also important to know the locations of where we store it. We store it either in the muscle. There's the majority of it's in the muscle, skeletal muscle, and then we have some in the liver. And the glycogen stored in the liver is responsible for maintaining our blood sugar when we're not eating much, when we're fasting. Cellulose is important in the body because it's fiber. That helps us to stay regular in our bowel movements, and also helps us with some other things when we're talking about soluble fiber. But the main reason here is because fiber helps us stay regular in our bowel movements, which is an important part of digestive health. Okay, and proteins we've already spent a lot of time talking about. We've talked about the primary, the secondary, tertiary down here, and then the quaternary structures of proteins. Okay, we know that they're made up of 20 different amino acids held together by peptide bonds, which are a form of covalent bonds. Okay, and we know that amino acid chains come together and they fold and form different shapes based on the properties of adjacent or near amino acids, and mostly these hydrogen bonds holding these together. Okay, so these shapes are maintained by hydrogen bonds and covalent bonds, denaturing of proteins due to high heat or chemicals or pH imbalances affects mostly these hydrogen bonds. So we're going to look at some examples of proteins that illustrate the different functions of all of proteins. Okay, so the one main function of proteins is the structure of the body. Okay, so one example of this is collagen. Collagen is your major protein in all connective tissue, and it helps maintain our structure. That meaning we find it in our bones, we find it in our tendons, ligaments, muscles, we find it all over the body, and it maintains structure. Okay, second function of proteins is movement. Okay, actin and myosin are these contractile proteins that are shown here. This is the myosin head. This yellow balls are the actin, and this is exactly how muscle shortening occurs by the myosin head pulling the actin and shortening it. Okay, so those are two important for proteins. Next, another important function of proteins is to be a catalyst. Okay, so catalysts are enzymes that are either essential for a reaction or they speed a reaction. Okay, so certain reactions don't happen at all unless there's an enzyme. Other reactions can happen without an enzyme, but they're much faster and more efficient if the enzyme is available. This one we talked about in class is the example of the enzyme heparin, helping thrombin to be inhibited to stop blood clotting. Okay, this black is what the heparin is supposed to look like. Okay, another important function is transport. Okay, so hemoglobin is an example of this, and hemoglobin transports oxygen to cells. Okay, so we find tons of hemoglobin inside of these red blood cells, and hemoglobin specializes in binding to these oxygen molecules and carries it down to the cells for use in metabolism. Okay, proteins are also important for pH regulation. Plasma proteins like albumin, which is your most common protein that's just found in your blood, can act as a buffer. Okay, it can act as an acid buffer or a base buffer because there's certain amino acids on it that are acidic and some that are basic, and they can soak up hydrogen ions or they can release hydrogen ions if needed to help us regulate pH. Another important function of proteins is regulation of metabolism. Okay, so some examples of this are insulin, growth hormone. Those are protein hormones that are responsible for dictating metabolism of other cells. And this is showing insulin which is a protein binding to an insulin receptor, which leads to glucose being brought into the cell. So that's helping regulate metabolism in that way. Okay. And then finally, the two other functions of proteins are one, body defense system. Okay, so antibodies are these little proteins here, and these are what the immune system creates to destroy invaders. So invaders being bacteria, viruses, funguses, different things like that. And your immune system makes specific antibodies for every invader that it's encountered. Okay, and those are made up of protein. And then finally, protein management. We have these things called molecular chaperones, which aid in the synthesis and breakdown of other proteins. So proteins are also helping other proteins be synthesized, just like the ribosomes, and also helping us break down proteins. Okay, let's move on away from proteins and talk a little bit about lipids. You guys have seen these lipids before. At least this phospholipid is one we talked about in class earlier in the week. And a phospholipid is important in the cellular membranes. Okay, so all lipids are made up of carbon, hydrogen, and oxygen. Okay, they have lower levels of oxygen than carbohydrates, and some have phosphorus, just like as in the phospholipid. Okay, these are insoluble in water. All right, three important types here are the phospholipids, triglycerides, and they're triglycerides because you have the three glycerol molecules back here, and then cholesterol. Which is what makes up our steroid hormones. Okay, so I will want you to recognize at least the triglycerides and the phospholipids. Okay, so triglycerides are considered neutral fats. They're made up of fatty acids and glycerol. Okay, in a 3-to-1 ratio. So the glycerol is the head, and then we have 3 fatty acid tails. Okay. So the main functions of these are energy storage, insulation of the body, and cushioning of organs. Okay, so we have fat around kidneys, around our heart, and that helps us to cushion organs. And insulation, obviously, that can help keep us warm and maintain temperature. And storage of energy, this is our main way that we can store energy is in fat. Very highly energy efficient and packed full of energy. Okay, we can either have saturated or unsaturated fats. Saturated fats means that there's single covalent bonds between all the carbons. Okay, the way to tell these apart, we know that saturated fats are solid at room temperature. So this is butter, this is animal fats, and it's found inside of meats. Okay, and then unsaturated fats are when we have one or more double covalent bonds. If it's just one, it's called a monounsaturated fat. If it's two, it's called more than one, it's called a polyunsaturated fat. And these are liquid at room temperature, and so these are typically found in plants. And this is our oils, our cooking oils and things like that. Okay, and I do want you to recognize the difference between saturated fatty acids and unsaturated fatty acids. And the way you tell the difference, saturated fatty acids are straight. Unsaturated fatty acids have a kink in the little tail. Every double bond causes a kink. Okay, and that's what you see here. This is a polyunsaturated with multiple kinks. This is a monounsaturated with one kink. You don't need to know the difference between poly and mono just as far as recognizing them, but I do want you to be able to recognize a saturated fatty acid versus the unsaturated fatty acid. Okay, phospholipids, we've talked a lot about these. We're not going to spend time on these because we talked a lot about them last class, but you can look at this and make sure you've got all this. Okay, and then finally, cholesterol is another very important lipid, and so this is a lot different than the other lipids. It has multi-carbon rings, and they're very important for three main functions: cell membranes, like we talked about, maintaining the structure of the cell membranes, steroid hormones, and then biosalts. And so dietary cholesterol is the cholesterol that we eat, and once had a very bad rap in increase in our cholesterol, but we've actually found out since then that actually eating a lot of cholesterol doesn't necessarily raise your blood cholesterol as much as you might think.